first promising session of the workshop titled Development of a Email-Based Method to Identify Infection Status in Whole Virion-Based Vaccinated Individuals by Professor Debasis Dash, Director ILS Bhubaneswar, Professor and Senior Principal Scientist, G. N. Ramachandra Knowledge Center for Genomic Informatics, CSIR Institute of Genomics and Integrative Biology, New Delhi. Professor Das has completed his MSc and PhD in Biophysical Chemistry from the University of Delhi and joined CSIR IJIB as a scientist. He has made significant contributions towards development of silico tools for large-scale comparative genomic studies, and some of these are extensively used in academics. He has more than 50 international publications, 2 patents, and several copyrights to his credit. He has been awarded the CSIR Young Scientist Award in 2004 and DBT Bioscience Award for Career Development in 2014. It's a great honor and an absolute pleasure to have him with us today. With a huge round of applause, please welcome Professor Debasis Dash.
Thank you. Am I audible to the last chair? I was wondering why people give so many blank places in between. Is it the clustering based on their friendship, or they think that it is better audible at the back seat than at the front? Anyway, it is your choice, but thank you very much for inviting me here. Let me tell you all of you that I am going to share some of my work which we did during the COVID lockdown time, which was very exciting to us. I hope I'll be able to share the same excitement to all of you. And no idea in the audience, those are there people over here are students, faculties, or a mixture of both. And also, sir, I'm told there are several online participants are there. So any questions, you can put it in your chat box at the end of the talk. I am ready to take the questions. But before I go further, I will also like to interact with you. Otherwise, this talk may become very boring and monotone. So how many in this audience understand the word whole virion-based vaccine, that is Covaxin? Whole virion-based vaccine. What does that mean? Anybody in this audience? So if not, then I will just little bit course change my topic in such a way that you understand. Otherwise, it becomes a monologue. Can anybody tell what is a whole virion-based vaccine or Covaxin? Whole virion, according to me, that you actually take the entire virus, kill it, or make it kind of inactive, and then inject as a vaccine. So as a result, your body's immune system elicits a reaction similar to that of a infection. In case of normal infection, the way your antibody will be developed, similar way your vaccine also exists. But there are other type of vaccines, for example, Covishield and all, which are mRNA-based vaccines. When they are injected to you, anybody who has taken Covishield here? Yes, few hands. Okay. When you take Covishield, then only S antibody will be developed in your body. And when you get naturally infected, then you get both N and S antibody. So we can easily say that the antibody level that are there within your body is because of the vaccine or because of the infection. So here is the question that got me over here. If you look at this middle one, Covishield induces only spike antibody, while Covaxin, which is a whole virion-based vaccine, induces all the type of antibodies such as N antibody, S antibody, and thus it is difficult to identify whether the protection that you are getting is because of the vaccine or because of the infection. I hope I have been able to set the stage how computational biology or a machine learning tool model is going to address a situation like this when the government is asking us, tell us if these people have been protected. Is it because of the whole variant vaccine or because they got infected and they got protected? That is the question. Apart from that, there are few other areas where I have worked, which I am not going to present today. One is that how a modeling-based approach was able to determine the covariates in susceptibility to acquire the virus. For example, we developed a machine learning model which could predict with certain accuracy level that who are more prone to get COVID and who will be protected. Looking at your blood group, looking at your travel, whether you are taking public transport versus private transport, looking at your age, gender, many other covariates, socioeconomic status we had taken. And by taking that, we could predict who all would be susceptible for getting infection. And the third story again, which I'm not going to present today, but these are all published papers. Can an AI tool be made which would behave like a radiologist and tell if the person who is infected with COVID need to be home quarantine or hospital quarantine, which means if the person has certain COVID pneumonia, the chest is infected with pneumonia, then you have to suggest that person to get hospitalized. But if the patient or the individual has just got RT-PCR positive, but there is no pneumonia that has set in a resource crunch system and the hospital beds are really low, then you advise the patient to get home quarantine and remain at home. So what does this tool do? This tool takes the chest X-ray through a knowledge-based system and AI model. It tells whether the individual is COVID likely or COVID unlikely, COVID pneumonia likely or COVID pneumonia unlikely. With 97% specificity on Western dataset and 86% specificity on Indian quarantine center dataset, the model was able to help people take decisions as to how to deal with this. Now, coming back to the main story of today, which we are going to discuss, is that how can we ascertain that Covaxin or a whole variant-based vaccine is giving us protection or not? Let us see that. To do that, what are the ingredients needed? How do we start a problem? I'm a computational biologist, a formally trained chemist, turned bioinformatician, and for last several years, I've been dealing with developing AI models and several other models which can be utilized for biological problems. I believe many of the people in this audience have an interest in that area. That's why I'm sharing this story with you. So the starting point of such a project was to actually have input data. What is the input data? Input data is from all over India. We had collected individuals' blood sample and also filled a questionnaire. Through the questionnaire, we got information about age, gender, number of vaccine doses they have taken, date of collection, date of first vaccination, date of second vaccination, and similarly many other questions we asked, and we collected the data through a questionnaire. Also, we evaluated their antibody level by taking small quantity of the blood of these individuals, whether they have antibodies against the spike protein and antibodies against the nucleic acid. So I will call it S antibody and N antibody for brevity throughout my talk. When I say S antibody, you imagine that this is against the spike protein, which is generally also elicited by Covaxin. Covaxin only gives you this antibody. When I say N antibody, this N antibody is generated because of the natural infection as well as Covaxin, but they also elicit this antibody. So the question is that a natural infection as well as Covaxin gives you both the antibodies. That's why the confusion starts, and can we provide a solution? Estimate the burden of SARS-CoV-2 infection, evaluate the vaccine efficacy. This is the goal. Would anybody try to give me a hypothetical situation here without feeling shy? That if such a problem is given to you, how as a computational person, you give a solution to a problem? Can we predict that whether the protection you are getting is because of the vaccine alone or the infection? And if so, what would you do? Would anybody like to give a try? If not, I mean, if you don't interact with me, it becomes a monologue. It will be a one-sided story. Okay, let us go ahead. So we had a background information such as, just remember 2020 July, 2021 July, and then 2022. These are the various infection we had. The wild type infection here, the Delta wave, and the Omicron wave. These are the number of individuals which got infected. And I'm talking to you about a story which got unfolded somewhere here. While the Delta wave was going on, we started this work, and we had collected samples at several stages. There are two types of terminology I will be using during the course of my talk. When I say somebody is infected, that means the RT-PCR status, which talks about active infection. Anybody have done RT-PCR? You must have done RT-PCR, right? It talks about active infection. That means आपको अभी infection है, the RT value is high, so that the virus is there in your body. But then after 15 days, if you do the RT-PCR, then it is gone. RT-PCR is negative. Does that mean that you were not infected? No, you were infected. The active infection is gone, but now the antibody has taken over. You have developed resistance. You have developed antibody, and the stage has come where you have been protected. So RT-PCR gives a quick and specific acknowledgement about the infection. If it is there, asymptomatic persons also may be missed because they will not go for the testing. On the other hand, serological assessment, and this is where the sample is that you take some nasal swab, throat swab, all these things, biological fluid, and then you get this RT-PCR. On the other hand, serological assessment, you take blood sample, and in the blood, you try to monitor the N antibody and S antibody level. The advantage of antibody level measurement is that it exactly tells you whether you have been infected, and right now the antibody level is whatever is the antibody level. But the difficulty is that to do this, you need the blood sample, you need the serum. So what we did during our study, we collected sample from about 10,000 individuals. 10,000 individuals all over the country and at different stages. For example, phase 1, June to September 2020, phase 2, which is January to March 2021, and phase 3, June, July 2021. And many of you can jolly well imagine when you do this kind of cohort-based study, then it may so happen that those people who participated here may not have participated here, may not have participated here. That means when I do a Venn diagram of who are the people who have participated in all three, that will be very few. Who are the people who have participated in at least 2 phases, it will feel larger. And the union of that was probably 18,000 or so. That means 18,000 individuals have participated, but those who have participated only in 2 or all 3 are fewer. All this work has led to several findings. One of the major finding was, when we are doing this study, is that 20 to 30% lost their ability to neutralize the virus in 6 months. That means they need to be given the second dose, and so on and so forth. government needed a lot of policies to be discussed and debated. So the work that we have collected this sample and study has led to many stories published in Nature Communication Science and again Nature Communication. These are all the wet lab side. I am going to talk to you something that a dry lab person like me and my students in collaboration with these wet lab people unfolded, which was possible because this data was available. So what did we do? This is a repetition of whatever I told in the beginning, that only N antibody level is seen in case of Covaxin. As you can see, this all the antibody level is on this axis. Very rarely there are samples on this axis. Whereas in the case of Covaxin, you see all over the place the sample individual. It's hardly visible to you on the screen, but then take it from me that people have developed both N antibody and S antibody because of this Covaxin. And the objective is to develop an artificial AI-based method to serologically identify infection status of a person administered with Covaxin. These are the various phases where samples were collected: phase 1, phase 2, phase 3, which is overlaid with the background information. The background information is about the COVID-19 confirmed cases all over India, and we are collecting samples in the red ones here as well as here. Now the question that we are asking over here is that these people, are they protected enough because of the vaccine or because of the prior infection? That is the main question that we are addressing. Remember that. So P1, P2, phase 1 and phase 2 data was collected before even the launch of vaccine. That means people have not taken vaccine during this phase. So they are either vaccine naive, or they have taken 1 dose, or they have taken 2 doses. The sample that I have, I've got all the 3 set of population. Either they have not taken, or 1 dose, or 2 doses they have taken. I hope you are with me and understanding the challenges. In such a scenario, this in one slide, in one picture, I'm trying to capture the entire sample set that I have and what kind of story the sample tells us. For example, if you look at this sample distribution, we have about 1823 samples who have 1823 individuals who have taken Covaxin. That means the rest of the people have taken Covaxin. These 1823 individuals, this is the status of their participation in various phases. This is phase 1, phase 2 and phase 3. The color code tells that the one that are shown in this brown color are seropositive. That means vaccine was not available. If they are seropositive, what does that mean? Vaccine तो लिया ही नहीं है, और seropositive से phase 1, phase 2 में, that means they are already infected. So for me, it is very easy to know I know their antibody level, I know they are seropositive and they are not vaccinated. So what is the level of their antibody with just infection alone? The rest of the people, like the blue one, they are very interesting. They are saying we are not infected. Self-declared not infected. What does that mean? I say that I am not infected. How do I know I am not infected? Yeah, that means I have not got any symptoms. But that is something called asymptomatic infection. That means I did not get symptoms, but silently got infected and developed the antibody. Now, Covaxin people are telling, no. They got antibody because of giving them the Covaxin and the antibody has left. But they have not taken Covaxin during phase 1 and phase 2. So, the confusion is that, are these people really not infected or they have got a silent infection? The gray ones are definitely not infected because the sero value is not there, antibody, they have not taken the vaccine, so they are definitely not infected. But what is the ground truth is the yellow color one or the golden color one, which are saying that I am infected. That cannot be ignored because they are saying that they are infected, they have been RT-PCR positive, and they have symptoms. So for me, the golden color ones are more or less a fact that they are infected. So if I have their antibody level at a certain height or certain peak, That means I know the antibody level reaches to this height when somebody is infected with 1 dose, with 2 dose, with 0 dose. I hope you are with me. If you understand this, much of my problem is solved. Rest on the computer application. So I take this as my learning place where I am saying they are all infected with 1 dose, 2 dose, or 0 dose. So this is a depiction of the antibody level of the S antibody and N antibody and how the people have been distributed, frequency of the samples, where this is a bimodal distribution. And the covariates, as I told you earlier, I'm just repeating, age of the person, gender of the person, number of doses they have taken, and days since the last vaccination. Most important is that if I have taken the vaccine 1 month before, 15 days before, 1 week before, or yesterday, that means the effect of the vaccine has not set in because I have taken only yesterday. If I get antibody very high and I say that I have only taken vaccine yesterday, make sure that these people have been infected earlier. That's why the antibody level is high. Vaccine has not given. But I may say that common sensically, is the computer is saying so. So these are the few factors we have been given written as the covariates, which can influence the level of the vaccine. What data is being collected or generated? Antibody level of both types, age, gender, and number of doses days since last. So if the trend is like this, the dose since last days since last vaccination, it is greater than 8 weeks. That means the trend. That means the antibody level will go down. Here is little bit complicated. Let me try to explain. I have taken vaccine 1 month before, 4 weeks before. That means it is still like increasing stage. But I have taken vaccine since 2 months or 3 months. That means the effect of vaccine again has gone down. So I need to understand that this value that I have, which is a saturation value, whether in this phase or this phase. So this since last vaccination helps in determining that. Now comes the job of a computational biologist, which I think plenty of you are there in this audience. If you look at the left-hand side, these are all the self-reported infected people, 375. That means the golden color or the yellow color. They are all sitting here with very high antibody level. You can see the epicenter of this data is this high. This is a log scale, and this is 2, and this is about 3. Spike protein 2 and antibody level. But interestingly, those who are saying that we are not infected, that is 1448, about 1500 people who are saying we are not infected, they are ranging from all the way from 0 to all the way here. You see a lot of dots here. I hope it is visible from far. There are a lot of individuals over here who are saying we are not infected. These people are also saying we are not infected, but they are behaving something similar to the infected. So can I now label these individuals as infected? Probably yes. But if I say so, then people will not believe me. So फिर क्या? आपने तो फिर पहले से assume कर लिया कि these people are infected and they may be infected. So I have to do it in a computational rigorous way, that whether or not they are infected. So I have to develop a model. So we split the data into again further subgroups such as infected, not infected, 1 dose, 2 dose. Now the data got little bit separated into 4 quadrants and the density of the data finally. Now look at this one. It was the overall story of not infected 1448. Look at this corner. They are saying we have only taken 1 dose, but still they have such high antibody level like the infected people. Now with 1 dose, people cannot generate so much of antibody. 1 vaccine dose. So can I take this as infected people? I'm tempted to say yes, but hold on. I don't want to. I want to tell my computer, my model to say that whether they are infected or not. What I do next, in a PCA plot, I look at the contribution of each of the variables in segregating this, where the infected as well as self-declared uninfected people are clustering together. Still not happy? Okay. Let us develop a machine learning model which will tell us whether these persons are infected or not. I will take few minutes here to explain you what does this model do. In fact, there is a supervised approach and unsupervised approach. In a supervised approach, I give them a label of who are infected and who are uninfected. So in the previous slide, if you look at it, I know for sure that these people are uninfected because they are saying they are uninfected, their antibody level is not there. So this is my negative set and this is my positive set. By taking these people as positive and negative, I want to ask a question whether these people are infected or not infected. So in the supervised model, in a 70-30 split of training and validation, we created a model which will predict through several iterations using SBM that what is that partitioning I should do where I say below this are uninfected and above that are infected. But this is a supervised approach. In an unsupervised approach, we took a prototype LCM based model and tried to create how many clusters are there in my data. Is it 2 cluster, 3 cluster, 4 cluster, 5 cluster, number of clusters, where sometimes we can say that number of cluster I dictate, sometimes we want the machine to decide the number of cluster based on the sellout score. But then finally we took a concordant label between the supervised model and unsupervised model of these 950 individuals, which is relabeling them that whether they are infected or not infected. So what we did only on this 965 out of the 1800 we had, whatever model we developed, which is an ensemble model, that we applied on the remaining data set, which were not used in the training or testing, absolutely blind, whether those individuals were infected or not. So these are a blind set from 965 to 1823. Whatever that remaining has never been used in training or testing. So this is the crux of the entire computational model, which is published in the CBM journal, where you can go to the details. But then let me tell you, after doing all of this, the reviewer was not happy. They said, okay, we want a secondary proof, an experimental proof that these people are not infected by the wild type virus. They must be infected with the Delta type virus. So can you show us, prove us that? To do that, what we did, yes, here. So when SBM or the supervised approach has segregated them as infected and uninfected segregation, and this unsupervised model is putting them in the top right corner versus bottom right corner by creating a partition, then we are saying that these individuals are always labeled as infected and labeled as uninfected. For example, to tell you again from this data, these guys who are sitting on the top floor, who are saying non-infected, they are labeled again and again as the infected guys by the model.
950 were detected by SBM that they are infected.
950 people were total taken into consideration as infected or uninfected. The labels were ascertained. For example, we had 370 people who are infected. This is for sure. We know that they are infected. We are not doubting their ability that whether they are infected or not. Now this 375, some of them now join this level, saying that they are infected. Now these people, let's say they are saying they are infected. Some of them may be uninfected. Because of the wrong testing or something, they are saying, or I got a symptom, and they say that I'm infected. So very few people were there. So coming back to your question, the concordant level between machine learning and clustering, this approach, this side and that side, where we get a singular level that they are either infected or uninfected, which is backed up by the data from the P1, P2 seropositivity. P1 and P2, let's say you are saying that you are infected, or you are, let's say you are saying uninfected, but I am sharing with you that phase 1 and phase 2 your seropositivity was there. Okay, so that means your level now has been converted from uninfected to infected. So this is what we got the final concordant level, but the testing is still done on 1820. There is a total sample size. Because many of them have not seen the model, they have not been exposed to the model, they are kept aside in the beginning itself. That are these individuals. We have never exposed them. But then, as you know, these individuals which are never exposed to the model, we know that their status, whether they are infected or not. Can I use this set as a ground truth? If my model is telling that these people are infected, I know that they are infected, that means model is working fine. If these blue people are saying uninfected, but some of them I am saying they are even infected, that means model is adding few more to the infection category. Okay? So, what we did, we took consensus of two unsupervised clustering algorithm that was I had shown in the flowchart before, where the label, the clustering is telling that this is cluster 1 and this is cluster 2. It is not talking about whether they are infected or not, because this is a computational technique which is simply clustering them. So this is cluster 1, the one this color, and this is cluster 2, the blue color. Whereas the supervised learning, which is 100-fold bootstrapped SBM model, is saying that these are infected and these are uninfected. It is now up to us to give this cluster a name. So far, this cluster has not been named. They are cluster 1 and 2. But after getting the supervised model, where this is uninfected, this is infected, these are all individual. Each circle is an individual. We are giving them a label. Okay, these guys is uninfected, these guys are infected. With the caveat of this color intermediate, that we cannot say about the status of these individuals, whether they are infected or uninfected. They are kind of a midline value. So if you look at it, the AUC under the curve was pretty high in our prediction system. Accuracy was reasonably enough, above 0.8, and this was taken further for more of the same thing which I presented in the previous slide. The 100-fold SBM level, infected, not indeterminate, and not infected, cluster 1, cluster 2, and indeterminate. So clearly, these people are clustering together, which are labeled as infected. These people are clustering together, which are labeled as not infected. So these are two, this 950 that you are asking, this plus this is the 950, which is now fed into the ensemble model as an input with their label as the infection status. The model that was developed now applied on the entire 1800 to predict the status.
So,
yes.
So we only use that
950
datasets only.
Hmm.
So that all the 18,000.
1800. Yeah. So this 1800, out of the 1800 something, 950 is now further taken to the model.
Then all the remaining datasets are applied for the
testing purpose.
Okay.
Of which, if you remember this graph, please remember this graph. This one, is it visible to you on this side? Here, there are some gold level infected people and some of them doubtful uninfected people. They are now being predicted. I am using these people as the last blind set where the model is working or not. If my system says that many of these gold people are uninfected, then I will doubt my system. because we have RT-PCR positive data, we have seropositivity data, but still the model is unable to predict that. So that is not the case.
In this model, in simple model, you have taken SPM, random forest, gradient, and KNN logistic resistance.
Correct.
The thing is that when you choose the eligible model, why you have taken this model? Why other models have not taken up in this?
In this ensemble model, why I have taken 1, 2, 3, 4, 5, 6, and not taken what?
Other models are there, like XGBoost is there.
Okay. So when I have taken 5 things, why I have not taken another?
What is the reason behind taking this 5 model? Is there any advantage available from these 5 models so that you have taken?
No. Actually, I would not have taken even this 5. It is like you have to prove in a computational way. XGBoost was also used to get the ensemble data as a whole, but then finally we reported this because you have to take more than 1, so you have gone for 5. You can
Randomly we will choose more than 1 or looking into the previous scenario.
no, not looking into the previous scenario. You take methods which are independent of each other in terms of their background.
Any method we can.
Yeah, you can take any method. So right now, these methods were taken to predict all the 1800, and whatever has not gone into this any of these model prior, their result is going to be reported that whether it is accurate or not. Not only the computational prediction, it has to be also validated using some experimental results. So here comes the most challenging part. So the ensemble model, as you can see, gradient boost is here. That you are asking. Sorry, XGBoost has not been taken. Gradient boost has been taken. So the majority polling has been taken to predict the output. Trained on these data points, the ensemble model identified 1,080 persons that are infected. Why I am insisting on infected? Because that is the one which I can prove again and again they are infected. The one that are not infected, it's difficult to prove because their antibody level might have gone up and gone down. So we do not, by the time we are checking, may not be that active infection is there, but definitely those who are infected, we can say that our model is predicting of them or not. This is the final validation. It might be very difficult for you to understand. I will come to you. Allow me 1 more minute to explain this before I take your question. What we did, we took certain individuals which we were saying that they are infected, but not by the wild type, but by the delta. That means the antibody that are there within their body is not because of the vaccine. That is the only one which, if I can prove, then I tell that this guy's antibody is not because of the vaccine, but because of the natural infection. And natural infection is not wild type, but delta. Are you getting my point? So if I can prove that point, that is the only way I can convince the critic that it is a natural infection. How do I do that? I do a computation. Computation between delta inhibition versus wild type inhibition. So we took their antibody and tried to neutralize the effect of the virus. If the antibody is because of the delta virus, it will neutralize the delta better than the wild type. If the antibody is because of the wild type, it will neutralize the wild type better than the delta. It is a common sense. So what we did, we took the antibody level of this so-called delta infected people, which we are saying infected and infected by delta. Their antibody neutralization was far better for the delta inhibition. So on the x-axis, while this is a scatter plot, so those individuals which are above this line, that means their delta inhibition is better than the wild type. Those who are below this diagonal line, their wild type inhibition is better than the delta. So quite a majority of our prediction, we could show that yes, they are naturally infected and the antibody level is not due to the vaccine. This is the coffin and nail of the coffin where we showed that outcome. model is able to identify whether the individuals are vaccine protected or they are protected due to infection. Yes, question. Sound loud.
Sir, I was having a doubt related to why we are not using K-means clustering. So we have some data sets.
Why we are not using K-mean? We are using K-mean.
So the thing is, we can group them like some properties are like we have one group which has more than one properties. We just need to cluster them, and after analyzing all the clusters, we can predict whether this is they have developed N type or P type.
Come again, please.
So my motto is to tell why we should use K-means clustering to group all the data sets, and we already know that some clusters have more than one properties, like they have one dose, and they are telling and their immunity level is very high. So if we clusters them
along with the infected people, the infected people as well as these one dose people will cluster together.
Yes,
we did that. We did that, but not by just TMN clustering, but by many other ways to say that these people are in multiple number of ways behaving the similar way that of an infected person. So there is a reason to believe that they were infected.
Are there any properties or any characteristics which are already developed related to this model? Like if we have some characteristics, that means we are already developed P type or N type.
Okay. Let me again explain you that there are multiple forces working on an individual when the antibody level is generated. Of course, everybody is not a robot. Their own immunity level also varies individual to individual. Factors that are influencing the antibody level are: you are a male or female? What is your age? When did you take the vaccine? कितना दिन हो गया जब हम sample ले रहे हैं आपके? We are taking a blood sample. Sufficient days have passed, or more than sufficient days have passed. If more than sufficient, so what we are measuring is one data point. That is your antibody level. I measured it today. I measured it after 3 months. I measured it after 7-8 months. Phase 1, phase 2, and phase 3. I have only 3 data points about you, and all other data that you have provided me through a questionnaire. With that, if you are saying I am infected, then I label your antibody level. This is the antibody level of a male who is X-year-old, having infection, sitting here. So many such people will contribute to that cluster who are definitely infected, and we know your antibody level. The question comes when you say that no sir, I am not infected, but you are sitting still with all other friends who are infected. I need to establish that no, you are infected. It's not because of the vaccine, it's not because of anything else. This I could do through multiple number of ways, all of them trying to corroborate the same thing in their own style. But still, people are not trusting this because it is again a computational way of proving the same thing in a circular algorithm. So we have to take some sample out. Then went for the experimental validation and say that, see, whenever you do experiment, and also world is not fair enough in biology that you get everybody on the one side. I wish there were no spots on this side. But there are people, a small fraction of the people here, but most of them are here. So with that, the argument is that our algorithm is predicting more than 80% accuracy or 90% sensitivity. That these are all people infected with a new virus, that is Delta. That's why the behavior is different. But this could have been done for all 1800. You can, some of you might argue, आप पूरे 1800 के लिए ये कर लेते, very expensive. And we are doing a postmortem here, by the way. What are we trying to do? Infection हो गया है. People have already gone back to their home. We are trying to assess whether these people are protected because of the vaccine or because of natural infection. What we could get from here, percentage of sequences classified as Delta during the surge is 75 to 80. We found a 71.8. when our method suggested 71.8, the ICMR result, which is body that you know, we were also doing a sero survey, they say 75 to 80%. We are more or less there. So that is another way of proving that whatever your model is working is correct or not.
So is the data set available?
Yeah, data set is available along with this publication in the supplementary set. But I am not done yet. I am trying to end my talk with something. Do I have time? What is that? I don't see a watch nearby. कितना टाइम मुझे दिया गया और कितना टाइम मैंने ले लिया? 11:50. I'm supposed to be going on till 12 or what? Go on. No. Don't be fair with me. Just tell that ये खत्म हो गया था. I can finish. Let me come to, is the data set available? Answer is yes. But don't spend time on this data set. There are better things to do, which I discussed with Sanjay Saxena in the previous thing. I'm looking for students, researchers, faculties to come in a crowdsourcing model to develop some other model which will be required by the biologists, which will be required by the life scientists. There are better ideas to work on. Okay, so that is what we can deal with. So let me finish this story. The summary of this is that Covaxin or the BBB-152, which is the alternate name, induces antibodies to all viral proteins, not just S antibody. Differentiating infection versus vaccine-induced immunity is difficult. That's why there is a need for a computational model. Hybrid ML model approach was taken, and 71.8% of the sample predicted by our ML to be infected were found to be delta infected by the delta variant SPNT, which is also corroborated by an independent study during that time, which suggested that 75 to 80% of the people have been infected with the delta. People who contributed, I mean, of course, I acknowledge all 10,000 individuals who have given their blood. Of the 10,000, the 1,800 people who have been vaccinated with Covaxin have been used here. Apart from that, scientist Shantanu Sengupta, collaborator, close friend from IGIB, he did a lot of work for the sample collection, sample analysis, the wet lab side work completely. Kumardeep is a colleague from the dry lab side, helped us in developing this ensemble model using various other different ways and which we have tried to prove the point. Dr. Viren Sardana is a medical professional, also is a collaborator in the CSR cohort project, helped in both wet lab as well as the dry lab side. These are the warriors, students, PhD student. Pratik is my PhD student. Rajat is the PhD student of Dr. Shantanu Sengupta. Satyarth is a PhD student. All are, these two are computer background people. This is a wet lab person. Satyarth works with Kumardeep. And big thanks to all the CSR cohort team, and countless people have contributed to this overall work. The series of 7-8 publications that has come from 37 laboratories. From each laboratory, about 10 to 15 people have contributed. A huge data source have been created. Data is available. Those who are published in this in their respective the additional data that we provide, that is obviously there. You can lay your hands on there. So with that, thank you very much for this part of the story. But when it comes to new ideas, new collaboration, we have developed a portal called cnrs.i3.in, which is a collaborative network recommendation system. The hero is sitting here among the audience. Vinith is a PhD student. Raise your hand, acknowledge. So we have developed a system where you come up with an idea or a new project. You place it over there. I showed it to Dr. Sanjay. Where is he sitting? I don't know. He is sitting in the front row. So he has seen that. It recommends that who all scientists should be collaborating to achieve this goal. And he has witnessed, you all can witness. All you do is that copy and paste abstract of any work, either your work or your guide's work or somebody, paste it over there. It will give you a recommendation of scientists within the DBT system, because the database right now has only 315 or 350 scientists. it will recommend who should you go to collaborate or take as a reviewer of your thesis or your paper, all these things. What is needed next? The future is this 350 has to be 3,500, 5,000, 10,000. The database has to be enriched. The model, if you are a model developer, you want to take pride in the model development. Develop open source model which can be plugged in, where we have certain standard results with us. If we perform better than that, can be plugged in. Or else, the third one is the curation. If there is any bio person sitting here, life science person sitting here, definitely one can read the article and say that, oh, this DLAI8 doesn't do anything with the empathy. Just machine learning has been referred, by the way, in his paper. Machine learning can be done for to do this, that doesn't mean he's an expert. So such kind of things can be curation. Curator's role is there. So anything that you think is part of your interest, you can catch hold of Sanjay and right?
Yeah.
Yeah. So he's willing to be interfacing the IIIT Bhubaneswar chapter. Right now, I have taken the owners of the DBT system, all the DBT institutes. But then the success of this story is that more and more scientists have fed in. For the time being, I'm being a little bit selfish of keeping it to only life science domain, keeping away the engineering. But later on, as and when we move bigger family, bigger, a lot of people are giving input. The computer doesn't mind getting more experts, getting more people into the database. The processors may mind. If it is larger than, I think we have created a GPU based code, right? A GPU, A100 you are using? H100 you are using, okay. So the system has been developed, there is a prototype standing there, but sky is the limit. We must do something first time in our country for our people, so that the funders, whenever they are distributing fund, there is a call going on, then automatically an invitation letter will come to you saying that, hey guys, you are an expert in this area, the government wants to talk to you, bring it on to board. So have this place more enriched with lot of information as in the background or in the model development. Thank you very much. I'm done. I'm ready to take questions online, offline, whosoever is there. Feel free to ask.
If anyone of you are having any questions, then feel free to ask.
Are there anything in the chat box? Can anybody have a look at this particular platform, Zoom or something else? So, एक बार chat box देख लीजिए, if there are any questions. How many are students here? 1, 2. Oh, quite a bit. So, do contact Sanjay if you see that you are interested.
Yeah, sure. Said no.
No, okay.
One question.
Yeah, please go ahead. Sound loud.
The chat window, there is one question. What about the status of asymptotic cluster?
What is the status of the asymptotic
Cluster.
So, I think these guys, they cannot probably see online my pointer over here. I can use the mouse maybe. Who are saying not infected here, wherever my mouse is. Those who are saying not infected, but they are P1 or P2 positive. The triangles. Look at the triangles. They are P1, P2, seropositive. These are the guys, definitely they are infected. Even if they say that I am not infected, my RT-PCR is negative, that means you have missed the window because you are P1 and P2 positive, seropositive. So these triangles, these guys happy because they say they are infected and we say also they are infected. But these guys, when they say they are not infected, we say, sorry, we don't believe you. You might have missed the RT-PCR test, might not have got the symptom, That's why you are asymptomatic positive. So we have no doubt about these individuals who are telling that not infected are infected. So asymptomatic, and that the question is, asymptomatic guys infected are sitting over here. We relabeled them as infected. We changed our label in the 950 are infected and trained the final model using them as infected. Thanks. Yeah.
So did you analyze
South.
So did you analyze any geographical relations in this data?
No.
This is an India level study.
Yeah, we did not. We did not put any because we did not have any hypothesis or really believe that South Indians are more prone to COVID than North India.
Like climatic.
Climatic might have effect, yes, but no, those information were not available to us. So monsoon is there, monsoon is going all over the country, or summer is there, but the temperature of course goes up and down. But climate and other kind of covariates have not been taken as input.
So we have seen the spike in July is the maximum.
Mm-hmm.
Is that rainy season having more?
Not really. I have no reason to believe that. All we know that 100 years of history of any pandemic that we have seen, it comes in spikes and then it dies down eventually. So, and the harmonic relationship that we are seeing, it is naturally. Only thing that people probably were not aware that Delta will be more dangerous because Omicron did not cause any damage. Infection was there, people got symptoms, they did not die. But Delta, the kind of mutation it harbored and the kind of damage is created was huge. Otherwise, this kind of distribution is .ibric.in. IBRIC.
Just write it down.
I can, provided. Can you write it down here or open a browser? Yeah, you got the question. Somebody please type open a browser. Open a browser and just write CNRS.IB.in. What is the question? Please read out.
So there is a question from Ayushka Chaudhary. Sir, how do we determine the window of the virus? And is it based upon the virus life cycle in a particular region, or it is compared with the virus of the same family, such in the case of SARS?
So, thank you for this question. I admit that I am not an expert of virologist. So I cannot tell you in general the life cycle of a virus, whether it will be 7 days, 5 days, 15 days or something. But in our case, in the coronavirus case, we used to see that mostly it survives in the bloodstream from 10 to 15 days. That means within the window, if you take swab and do a RT-PCR, then you get the signals of amplifying those DNA of the corresponding to the virus. But that is for just the coronavirus. When it comes to other virus, my virologist colleagues can answer, or even Google can answer. It can range between somewhere 5 days, 3 days, 5 days, 7 days, 10 days, 15 days, something like that. Coronavirus, I think we knew that it can last up to a month. There are many people living after 28, 25 days of the infection, active infection, they went for RT-PCR. They found the CT values to be positive, still positive. So that means the virus is circulating in your blood, in your stream. I hope I have answered. The CNRS.IBRIC.in. Hopefully it will open, otherwise we need. Yeah, I think it is coming up. So this may look very unromantic at this point of time because it's a proof of concept site. Why don't you come and give a demo quickly? Copy paste one of the abstracts I'd love to
Sir,
have your opinion on this.
sir, like efficacy, is there any side effects, study on side effects of that plant?
Plenty of study on side effects are there. But the most difficult was the efficacy, where virus will behave the same as that of the vaccine. Then how do you know whether you are getting a protection because of the vaccine or because of the virus infection?
Is there any comparative study between the Covaxin and Covaxin?
Yeah, plenty of studies. One of the people is myself, where we have Covishield was much easier for us because those individuals who got only spike protein and no N antibody, S antibody and no N antibody, we immediately labeled them as vaccine effect. Those we got both N antibody and S antibody, we immediately labeled them as infected. So once you get these two states, then immediately doing a small maths, you get the vaccine efficiency. All we had to have is their antibody level measured over a period of time. Yeah, copy that. So I paste it over here. So, so what we have done, we have taken Dr. Sanjay Saxena's one of the paper and their abstract. If let's say he is starting this work today, he decided that I will work on a project which is as written in this abstract. Now he wants to know किसके साथ collaborate करूँ, किसको चिट्ठी लिखूँ, whom do I, or I want to send this for a review, who would be my reviewer? In such a scenario, if you come and paste over here, I think this is done. So these are some of the scientists, and not only it is telling the name of the scientist, it is also giving you on the basis of what keywords those scientists have been chosen. If you go further down, the last bottom of this section, last or so these are some of these keywords which are contributing heavily. This is a translational science, diagnostics. If they want to take the better one, you should have taken the one that you have just now shown. एक तो लिया था पिछली बार नहीं. So he is talking about artificial intelligence based hybrid deep learning models for image classification.
Prediction is based on the context. So the larger the text we take, the better we get the mic.
Yeah. So is that my mic is on the podium? Just paste it and then while you are talking. Go for the straight one or maybe. स्ट्रिक वाला लेंगे या कौन सा लेंगे?
सर स्ट्रिक
ले लूं. And the number of scientists you can choose to be 5, 6, 7, 8, 9, 10, as many number of one you want. And this is a NLP based method, and for that top of which I need to explain whatever we have used word system.
Actually, in this project, we have used an LLM. So it is a part-based project. So it was basically created by Facebook, then they made it open source. So basically, it is of two parts. Like it is two parts of the word part and the GPT part. So what we are taking, the context from the paragraph. From this paragraph, we are taking the context and we are mapping those with the expertise of the scientist that we have.
So now the result has come. You go ahead. is not responding at this point of time. पर वो तो कनेक्टेड नहीं है ना लेवल से. Anyway, I think we are done. Proceed with the rest of the program that you have. I don't want to steal the show completely, but this is one area where we are looking forward to participation. And our purpose is that if you want to do something innovative, new, this is where. Thank you very much.
Thank you, sir.